Motility. Motility is the ability of an organism to move by itself by means of propeller-like flagella unique to bacteria or by special fibrils that produce a gliding form of motility. Motile bacteria move using flagella, which are generally thread-like locomotor appendages extending outwards from the plasma membrane and cell wall. Either it can be a single flagellum or it may be a multiple flagella. So the flagella are long filamentous appendages that arise at the level of the cytoplasmic membrane and extend through the cell wall into the surrounding medium. They are generally found in round shaped, which are motile, and they are generally found in gram negative bacteria and gram positive rods. For example, in Listeria and few cocci like Enterococcus species or Vagococcus species. So the ultrastructure of flagella on electron microscope, the bacterium flagella is composed of three parts. The first part is the filament, which is the longest portion of the flagellum that extends from the cell surface to the tip. Next comes the basal body, and the third part is the host. The basal body consists of M ring that is anchored to the membrane, S ring that is anchored to the periplasmic space, P ring which is attached to the peptidoglycan, and L ring that is attached to the lipopolysaccharide outer membrane. So the flagellated organisms alter the expressed antigenic type of flagella that they produce, and this phenomenon is known as phase variation. Based on this phenomenon, the organisms can be differentiated by the number of flagella, that is Monotrichous, for example Vibrio, Pseudomonas and Campylobacter. Lophotrichous, example Spirillum. Peritrichous, example Salmonella typhi, Escherichia coli. Amphitrichous, example is Alcaligenes fecalis. So this picture shows the different flagella. The detection of the flagella can be done by two types of methods. That is by direct demonstration of flagella using tannic acid staining and electron microscopy, and indirect means by demonstrating the motility, that is by Craigie tube method, hanging drop method, semi-solid medium, for example mannitol motility medium, and dark ground microscopy or phase contrast microscopy. So the direct demonstration can be done by electron microscope. Next, the indirect demonstration of the flagella can be done by hanging drop method. This is the most common method which we perform in the labs for motility, that is hanging drop method for bacterial motility. The hanging drop preparation is a special type of wet mount in which a drop of medium containing the organisms is placed on a microscope, often used in dark illumination to observe the motility of bacteria. This setup is the hanging drop preparation, where we have a depression slide, a cover slip and petroleum gel. So in the subsequent slides so the materials what are required for hanging drop preparation are glass slides, that is glass slide with a depression, or normal glass slide with adhesive or paraffin ring, paraffin wax, inoculating loop, cover slip, microscope, Bunsen burner, and young broth culture of a motile bacteria. You can, for example, Proteus mirabilis. After the preparation, we have to place a drop of suspension on the cover glass and invert it over the cavity slide or hollow ground slide. So the observation, first we need to differentiate the motile bacteria from passive drifting and Brownian movement on spot. The Brownian movement are the vibratory movements which are possessed by the all small particles suspended in the fluid. We need to focus the slide in low power, that is 10x objective, and high power, that is 40x objective, for observing the motility. Next method is the Craigie's tube method. In this method, we take one tube, outer wide tube which contains a soft agar, and an inner narrow open-ended tube which contains soft agar of 0.2%. Next step is a culture of Salmonella typhi 901 non-motile strain is inoculated into the inner tube. Then the motile strains which are present in few in number will move from the inner tube and spread to the agar of outer tube and form a film of growth on the surface. Next, what we do is we do the subcultures which are made from the agar surface of the outer tube, which yields motile strains. So this is the Craig's tube demonstrating the reviving of the motile strains from a mixture of motile or non-motile strains of Salmonella typhi. The next method is using a semi-solid agar. This is an indirect method. It will the principle of this method is to determine if an organism is motile, that is flagellated or not. The media which we use is composed of gelatin, beef extract, sodium chloride, agar, and it has a pH of 7.3. First, we need to inoculate the test organism in the media by stab culture up to 1/3 to 1/4 inch in the middle of the tube and incubate for at 35 degrees Celsius up to 7 days. If the motile organism will spread out in the media, then it is positive. If it remains in a site of inoculation, then it is negative. Next we can use is a flagellar stain, that is by wet mount. In this method, it is useful when the number of arrangement of flagella are critical for identification of species of motile bacteria. So the procedure is we use a small drop of water which is taken on a glass slide, then dip the sterile loop into the sterile water, then touch loop full of motile cells to droplet of water on the slide and cover it with a glass cover. We have to examine under high power, that is 40x. If motile cells will present, leave at room temperature for 5 to 10 minutes. Then apply 2 drops of Rue's flagellal stain to the edge of the cover slip. And after 5 to 10 minutes, observe it under oil immersion field in zone of optimum stain concentration. So we'll see Escherichia coli, if which has flagella, if we are preparing the mount of Escherichia coli. Next, the various types of motility which are shown by the different bacteria. That is, tumbling motility shown by Listeria monocytogenes. Gliding motility shown by Mycoplasma. Stately motility shown by Clostridium. Darting motility is shown by Vibrio cholerae Campylobacter. Swarming on plate is shown by Proteus. Corkscrew, lashing, flexion, extension motility is shown by Spirochetes.